Okay, the next step is metaphase. Metaphase is the second step in mitosis. Okay, here the spindle fibers grow from the centrioles and attach to sister chromatids, which are also called double-stranded chromosomes. I told you there's lots of names that all mean the same thing. Chromosomes. Okay, and they attach at the centromere, not willy-nilly, right in the middle. Okay, and the sister chromatids align along the equator of the cell. Okay, so let's draw that out. Again, cell membrane. Don't even worry if your cell doesn't even look like it's the same shape each time. Totally okay. There is no longer a nuclear membrane, so we don't have to worry about that. But I'm going to draw my centrioles. Okay, and I like metaphase because M for middle reminds me that my sister chromatids are now lined up in the middle of my cell. So here's one set, and here's another set. If we were in class, we would actually act out this whole thing, and each of you would be a different organelle, and it's kind of fun. Okay, so there are my sister chromatids, and I'm going to add my centromeres. So my spindle fibers have something to attach to. And now I'm going to take my spindle fibers and attach them to my centromeres. So we have one that attaches to each side. Okay, and normally there would be way more chromosomes or sister chromatids in each cell, but it would be a lot of work and more difficult for us if we tried to draw all 46 chromosomes, so 3 is more than enough for a good example. Okay, so now let's label this. Okay, the big points that are happening here. Well, let's look more at that one. Okay, spindle fibers attach to centromeres. Okay, so that's the one big point. Second one is sister chromatids line up in the middle. Line up in the middle. Okay, and that is metaphase. Okay, so we've done prophase, metaphase, now time for anaphase. Okay, anaphase means away. Ana means away. Okay, so away, anaphase. Haha, that's how I remember it, right? This is the third step in mitosis. Each centromere, that's like that belt buckle that's holding the sister chromatids together, divides in two. The spindle fibers shorten and they pull and separate sister chromatids. Okay, identical chromatids move to opposite ends of the cell. Okay, here we go. Ready? Time to draw our cell membrane. Here we go. Okay. Okay, so I got my cell membrane, and then I'm going to have my centrioles on each side. I think of them kind of like a puppet master. And the spindle fibers are like the puppet strings, and they're orchestrating the chromatids around the cell. Okay, now each of my chromatids has been split in half, right? So I have one, and when I draw the chromatids, I normally have the top part is smaller, and then I have a longer bottom part, and that'll come in handy when we're in my meiosis. Okay, and then I'll have another one here. And then I have my third one. Okay, so now that centromere has been split in half. So we use centromeres to count how many chromatids there are in a cell. So even though you might have double-stranded chromatids, you're only counting how many centromeres there are, okay? And there's another Amoeba Sisters video that talks about that, how to count the number of chromatids in a cell, and I will add that, okay? So now I'm going to have my spindle fibers. Look at, oh, they're so much shorter than they were before. See, look, doesn't matter that we're not in class, you still get my same little antics. Okay, so here we go. Now let's label what's happened here. Okay, so my spindle fibers, shorten, okay, and my sister chromatids separate at centromere. Okay, and that's anaphase. Okay, and the last step in mitosis, but not the last step in the cell cycle, is telophase. Okay, this is the fourth step in mitosis. Okay, so here a new nuclear membrane develops. And nucleolus form. Our spindle fibers disappear, our chromosomes uncondense, back to spaghetti or back to threads, okay? They go from being nice and short and thick and easy to see back to very thin and difficult to see, okay? Our cytoplasm divides in half. Okay, and the cell membrane begins to pinch at the center. And mitosis ends. Okay, so let's draw what that looks like. Okay, now our cell membrane is not kind of an oval, but now we're going to have kind of this pinch in the middle. Okay, and we're going to have our nuclear membrane starts to come back. Okay, and we have a new nucleolus. Okay, and then we're going to have our chromatin. Okay, now they're called chromatin, not chromatids anymore. So now we're going to have our uncondensed chromatin. And don't forget the centromeres. This part of mitosis, to me, always looks like very sad or very tired eyes. Okay, and we have our centrioles, one on each side still, but all the spindle fibers have been retracted. Okay, and all the organelles would have also duplicated. They'd also split in half. There'd be mitochondria on both sides, vacuoles, smooth and rough endoplasmic reticulum. Okay, but the big thing about this is what's happening with the DNA, okay, and our chromosomes, and that's why we're just focusing on that, okay? So that is the end of mitosis. Let's label what's happening here. Okay, our nuclear membrane reappears. Okay, we have no more spindle fibers. Okay, we have our, right here, cytoplasm begins to separate. Okay, so this is the beginning of cytokinesis happening. Cytokinesis kind of overlaps from telophase into its own phase. Okay, we have our nucleolus reappears. And we have our chromatids uncondense to chromatin. Okay, so from dense to noodles. Okay, and that is telophase.